Let's talk about data. I'm Salvador Gezan, and I'm with
Vanessa Cave.
So today we want to talk about power and power calculation and some of the intricate details that goes in there. And Vanessa, I want to put you on the spot. Can you give us a brief definition of what power is?
Okay, so I might just take a wee step back and just describe where power fits into hypothesis testing. So we've got a hypothesis testing scenario. We have two kind of mutually exclusive statements. One is the null hypothesis and one is the alternative hypothesis. And from our hypothesis tests, we kind of measure the strength of evidence against the null. So there's four possible kind of outcomes or decisions. So the null hypothesis can, of course, be in fact true, and we can reject it, which gives us the size of the test. So that's a false positive. It could be true, but we could fail to reject it, which is a correct outcome. Or the null hypothesis could be false, and we could fail, and we could reject the null hypothesis, which is the correct thing to do. And this is the power of the test. And the other outcome is when the null hypothesis is false and we fail to reject it. So I think I messed that up.
Yes, that's right. Yes. So the power of a test is what's the probability we have to reject the null hypothesis when in fact it is false. So making the right decision of finding, I'm thinking about a t-test, finding the significant difference on the two treatments when in fact that is true. So we made the right decision.
Yes.
That's good. And I'm aware this is a very interesting and relevant topic in animal science and in human science. So maybe you can tell us how you use it in animal science, where you have more experience.
Yes, so one of the things with animal science is often to do trials involving animals. It's a legal requirement. You perform an animal ethics assessment. And a very important part of this assessment is a power calculation. So this is looking at how many animals you need in your experiment to detect an effect of a given size or larger at a set power level. So typically in animal science, we set our power level to 80% as standard. We work out what our minimum difference that we're interested in detecting is. We get some information about the variability of it, our key response, and from that we calculate the number of animals that we need in our trial to give us a reasonable chance of detecting a difference of that size, if in fact that's what the difference is.
So I see what will be the minimum number of animals that you have to submit, or you have to kind of affect with this treatment or these two treatments in order to detect that difference. And I see the same thing in humans in some studies. I think the concept comes from there, where you don't want to have too many people being subject to what it could be a drug with side effects. For example, that you don't know if there are side effects. You want to minimize that number, that risk, but you still want to do have sufficient replications, in this case, sufficient sample size in order to be able to detect. And this is what I find very interesting, because if your power is too low, that means you're doing an experiment and even risking people or risking animals to a given treatment, and then you have not enough power. And
Yes.
so this is where I think about that 50% power, which is you have 50%. I like to use the analogy with the students. You have a student doing a project, and then you tell them the power is 50%. That means you have 50% of being successful in finding what you're looking, and 50% that you will not be successful.
Yeah.
Given that the differences are there. So I always find that very risky, because 50% is like throwing a coin, really. You need some more relevance than that 50% to make sure it's worth conducting the experiment. I mean, this is without even talking about the waste of resources and time and etc. So you mentioned 80%. That means you give yourself 80% chance of finding that difference. And why not higher or lower than that? What's your take on that?
So it's interesting. So 80% has kind of become the standard in a lot of animal ethics power calculations. And to be honest, I'm not really sure why that is. So when we do that, I do encourage the people I work with to think about a power that is appropriate for their experiment and the goals. But of course, if we increase the power by to a very large number, say like 99%, we're going to need a massive sample size, and that will probably let us detect very, very small effect sizes. But we've got to ask ourselves whether these small effect sizes are actually of practical relevance. Yes, and of course you don't want to go too low to not give you much chance of actually seeing what you want to see.
Yeah, and I like to think on the same lines on the competition between what is the type 1 error and the type 2 error.
Yes.
So the alpha level that we use. So if we are very strict with an alpha, that means we have a significance level, let's say instead of 5%, 0.1%. That means your power on the same lines is going to get reduced because you're being very strict to let something to be significant. Well, in this case, the null to be significant, to reject the null hypothesis. And so you have to find a balance between what's a good alpha for a type 1 error and what it's a good power in order to be more or less on the same lines. So
absolutely, yeah. So power and size of the test are related. We can't disentangle them. So if you increase one, you affect the other, decrease the other. So yeah, we have to think about the two things in tandem, really.
And so when we talk about the increasing the sample size as one way to increase the power, can you think about other ways to increase the power?
Yes. So another way to increase the power is to think about ways you can decrease the variability or the unexplained variation. And in an earlier blog, we talked about a technique called blocking, and this can be a very effective method of reducing the unexplained variation.
Yeah. And that reminds me also, when you see some of these studies on animal or humans, you have covariables, you have groups or contemporary groups, and all those elements that are considered. Now, one of the things I find a bit complicated is when you do the power analysis, and you have to come up with these a little bit of crazy assumptions. So you probably have experience with that. So what is the difference I consider to be reasonable, and then which is one of the main inputs on the calculation of the power, but also what is the background noise, which we were just talking. And you are doing all of this before the experiment. So what's your thought? What are your thoughts on this?
candid conversation with your domain experts, you can get a pretty good handle on what kind of the minimum effect size of interest is. Because, you know, because they're experts in that field or in that area of research. So you can just kind of come up with an agreement of that through discussing with your colleagues there. Getting an estimate of variability to put in your power assessment can be a lot more challenging. Sometimes you're lucky in that there is pre-existing related or relevant data sets or research. Could be
like
a published paper.
Like a preliminary study or
information published on your key response variable in a related scientific study. Or you may have some data from another trial that's relevant. Other times you do need to do an initial kind of pilot study to get that information. Of course, when that comes to animal research, that does mean you're therefore using more animals, but you don't necessarily have to get that measure of variability from your treatment group per se. You can just study your, you know, the control group and you know, a group.
That's what I think. I mean, if you have information on our paper and you have access to the raw data for controls, it doesn't matter what the other treatment is. You can always use the variability of the control as your reference.
Yes, as long as you can make the assumption that the variability is going to be similar between the treatment and the control group. And sometimes you might expect that it'll be larger in the treatment group, and therefore you do need to up your sample size. If you expect it's lower, then kind of like it's not so bad. Yeah.
You are listening to Let's Talk Data, brought to you by VSN, a world leader in the advancement of big data software solutions for the agriculture and bioscience sector. Let's Talk Data.
Yeah, and I noticed when I have done sample size calculations that I end up having lots of different scenarios.
Yes.
So like a large standard MSE or background error, and intermediate, and then a small one, and the same thing with the differences.
Yeah.
And you end up with these tables where you have like 2 entries and then different values of power for different sample sizes and so on.
Yes.
And it's not an easy decision at the end because I know this is very subjective. First, starting from what are the levels, then what are the assumptions of this variability or this difference. So sometimes I tend to wonder if it's really useful or not.
Yeah, I think it is useful. It gives people a feeling or understanding of the sample size that is likely to be required. But saying that, I think it should be taken with a bit of grain of salt, and you should also consider other aspects, like you know, what happens if your assumptions aren't quite correct? What happens if the variability is larger? The other important thing in animal trials that we think about is what happens if something goes wrong. So what happens if animals have to be removed from the study for reasons that might be unrelated at all to the treatment, but there are many reasons, particularly on farm experiments where animals disappear from your trial without your control. So you need to build in some like safeguards as well to make sure that when you do the experiment, at the end of it, you're going to have sufficient and reliable data to draw valid conclusions. So I'd say the power analysis plays into that, but there's many more considerations at work too. Yeah,
that's good. Yeah, I remember seeing some analysis where we tend to think originally 8, 9 maybe even 10 samples per treatment we think will be enough. And then we
Yeah.
do the power calculation and you realize, no, it's 15, 16, 17.
Yeah, it's a different order. Yeah.
And then you say, okay, so it's actually mostly double the size that we need to get
Yes.
for that power. And so, yeah, so it's like you say, you have to take it with a pinch of salt, and we have to kind of see if it's.
Think about it.
Yeah. And I wanted to touch into two more topics, but one of them is more related to what you said about other unforeseen things. And I always keep thinking about the relevance of potentially doing some simulations. If you have a better understanding of what could happen, then maybe you can simulate data, obviously under some assumptions, and then that will tell you a little bit, let's say your two distribution of your treatments between the animals or the groups, how they are going to be, how separated they're going to be. And I think we tend to underestimate that simulation work can help a lot.
Yes, yes.
And a bit like what you said, if for unforeseen things, you can actually even simulate certain proportion of the individuals that are going to drop out.
Yes, and when they're going to drop out, yeah.
The only thing I tend to think is some of these power calculations are rather difficult in terms of the software you need to use, and the mathematics are not very easy.
Yes, no, they can be very, very complicated. And so sometimes you can simplify your power calculation down to think of it in terms of like a t-test, which makes it easy, even in kind of complex scenarios. You can do that. Yeah.
Mixed model framework, but you say, I'm
Yes.
going to assume I'm just doing a t-test.
Yeah. So you can, but there are also excellent, packages and procedures for doing, for calculating power on more complex data or trial designs. And some of this uses a simulation approach. Some, yeah.
Well, I'm just thinking on so if you have a longitudinal study, obviously you want to compare your treatments, then you have repeated measures, correlated structure.
Yeah.
And also your hypothesis might be finding the significant difference at time point 5, but maybe at time point 3 also.
Yeah.
So you just have a bit of a more complicated process, and then you have to simulate. And also the other thing is what I've seen is when your test is not a very straightforward test. So if we're talking about a t-test under normal assumptions, it's fine. But then I also think, what about if you're going to use a non-parametric test?
Yes, yeah. And it gets a lot more.
It becomes a bit more complicated. Yeah. But I think the importance is there. I mean, it's like we say, you want to do an experiment, and you want to make sure you have sufficient replication to justify that the experiment is carried out.
Yes. So yeah, and well, again, talking about animal trials, we're interested in that we've got sufficient replication, but we also want to minimize the number of animals that we use. So we want to make sure that we don't have, too much replication with kind of wasting animals in a way by having far more replication than we actually need. Yeah.
And so then that brings me to the other thing I had in mind, which is what happens when you have too much replication. When we have, because obviously as we increase the replication, the way I like to see it is that you make the distributions of the null alternative narrower, which looks like they are more farther apart. But what happens when your replication is extreme?
Yeah, well, you have a lot more information, obviously, and this will enable us to detect smaller and smaller effects, which is both a good and I guess a bad thing, because sometimes these small effects aren't actually of any relevance and in a practical sense, that they're so small that they wouldn't actually matter in the real world. Yeah.
So then in that case, we are thinking that very large sample sizes will almost be able to detect
Yeah,
any difference.
Yeah, as your sample
So
size grows to infinity, that definitely better to detect something. Yeah.
I always like to think like let's say somewhere like Amazon, and it compares the trades that it does, let's say on January compared to February.
Mm.
Even if the difference are only $1,
Yeah.
because
Yeah.
of the amount of transactions that they do, which
Yes.
probably is billions or millions of transactions, then obviously they're going to find a significant difference for
Yes,
an
they are.
irrelevant difference in terms
Yeah.
of sales between the 2 months, you know. So yeah,
And I
so
think
let's,
yeah. Sorry, I was just going to say, I
no,
think this highlights also when you're reporting your results from like for instance a hypothesis test, you don't just report the p-value. Actually provided a description or a summary of the effect size and the uncertainty around that. And I personally find confidence intervals is a very efficient and nice way of, of doing this.
yes, in the can. Have you heard about doing power calculations after the experiment?
Ah, yes, yes. And it does come up a few times in some of the journals I read on animal welfare and animal behaviour, that it seems just commonplace to do a power analysis after you've done the experiment as well. And I always, I'm a bit unsure about these. I don't really understand the value of it. Mostly it seems that people use this post-experiment power analysis to, in a sense, justify why their results aren't significant and that the original hypothesis is perhaps still true. Saying that, these post-hoc power analysis are useful if you're then going to move on to do another experiment, kind of looking at the same thing, because now you've got a pre-existing data set that you can base your sample size calculations on. Yeah.
So
Sorry?
if I understand right, is this where you see this post-study calculation of power? It's when somebody finds that their results are not significant, but then they say, well, my power wasn't very high,
Yes.
so probably I couldn't detect any difference anyway.
Yeah.
So I'm just telling you that maybe the non-significant differences I found are significant because my power wasn't very high.
Yeah,
Yeah,
and I
but
think that's bad.
no, I don't think it makes so much sense, because obviously it could go either way. It could be that you
Yeah.
don't have, um, yeah. I mean, even the thing. One of the things that I tend to think a lot is sometimes you see these studies where there's huge differences between the two treatments, biological differences,
Mm,
but then the sample sizes per treatment, let's say, is 3.
mm,
And then you say, well, I mean, I see your difference. There's a trend. The p-value is not significant, but also your power probably is going to be less than
yeah.
50%. So you're saying, well, it is there, but you kind of fail to do the proper experiment with enough replication to find this difference that you wanted to. So I
Yeah.
tend to blame more the scientists to say, well, you did a study where you clearly didn't have
No enough.
enough
Yeah.
power, even though you knew there was an important difference to detect. So it seems
Yes.
like a bit of a waste of resources.
Yeah. Yeah. Definitely.
But
Waste. Yeah.
well, no, yeah, that's a very interesting. It's an interesting topic. I
Mm.
don't personally enjoy too much doing power size calculations. I find them a bit tedious. It's not doing the data analysis and calculating, but it's very important to justify a given study and so on.
Yeah. So because I'm mostly involved in it, power analysis is part of the NanoWet ethics application. It's at that stage before before the experiment's kicked off that you get a real good chance to talk to the scientists involved about the design, about what their aims are, and to influence that. So and the sample size power calculations are a nice part of that. So I actually quite enjoy them.
Good.
Yeah, but most
in plant breeding, it's not power calculation per se, but often, particularly, or even in animal breeding, also in aquaculture, we tend to think about sample size more like how many individuals do I need to have a reasonable estimate of the mean of that that treatment or that variety or that whatever. Not thinking about a comparison. So this is kind of funny because there is not about the null hypothesis comparing this against there. It's just kind of like in relation to the width of the confidence interval, really.
Yes.
Which makes the calculation of power a little bit simpler because you just worry about the n, and you have to have a t value and a sigma or s estimation, s square. But that is a very different concept. So there's no like power. So it's what.
Yeah, the width, your margin of error almost, the width of your confidence interval you're interested in. So it's all about the precision of the estimate. So making sure you've got enough information to make your estimate as precise as you require. Yeah, nice.
Exactly. So good. So it seems like we have touched over these last few sessions all these elements of replication, power, blocking. So it's very interesting to make sure we have the experiment all in order and we use the available tools.
Yes.
So, well, it was nice talking with you today, and I'll see you next time, and we'll see each other next time. So take care.
Yeah, perfect. Okay, see you.
Bye.
Bye.
See you. Bye.
You have been listening to Let's Talk Data.